And we did have a couple of Q&A questions come through. So I do just want to give a look at some of these. I know, Melanie, you had touched briefly on the double-strand RNA yield for regulatory organizations. I don't know if any of you want to comment on any tips or tricks you've learned in your interactions with the agency as it relates to double-strand RNA. I know that tends to be the hot impurity that everybody is kind of curious to learn more about today. Does anybody have any thoughts?
Yeah, I'm particularly interested in hearing your opinion on the double strand.
Yeah.
So the double-strand RNA control definitely is very important, right? So the immunogenicity or reactogenicity can be related to that. So that's why, but from the vaccine, and I think there's concerns on the therapeutics as well, right? So the levels of control might be different, and even for the company, and start with get the data, clinical data, to see what that level should be. That's very important as well. So basically, I think it's really how we control it at which level. It's really related to the data. I think even from the product, even within the vaccine, from product to product, maybe there's some variation as well.
Yeah. There's no real hard and fast limit yet, right? It's kind of dependent on the totality of what your data is telling you. Any other thoughts, Oriana or Melanie? Yeah,
that's what I would say. Like, I mean, it is the big question on everyone, and like, but what I will say is it all depends on your dose level. It all depends on what, like, when we work with this impurity, right? So we spend more effort on IVT process optimization to minimize the double strand, as opposed to then trying to worry about purifying it out later. That just seems to be an easier challenge to face, to do that. And definitely analytically it has its challenges. People don't like dot blot, but dot blot's more sensitive, but the ELISA is more quantitative. They both read out different scales of materials. So it's an unknown, plus the analytics aren't great. And so it's just something, I guess what I find is I haven't had any sponsor clients who have been tripped up with this in terms of their regulatory filings. They've just done their diligence to try and quantitate it and control it to as low of a level as possible. And they haven't, that's been successful.
That's a really good point. Yeah. Focus on your upstream.
So another thing is that depends on the utility as well, right? When we're talking about the cancer vaccine, I think some double-strand RNA may be helpful, for the immunogenicity.
Yes.
It's really, I think, I know we try to have a generalize the, the level, but it's really case by case in some situations.
It's so hard. Especially too, we aren't just working with different molecules, right, and different indications and dose levels, but we're also working with vaccines where you do want that sort of immune response, right? And you're working with therapeutics where you don't. So it's a head scratcher. Oriana, did you have any comments at all, or should we move on?
I think, like I said, the team is in the right direction there, right? Really instilling that quality by design into our processes, so that we are controlling our purities as much as possible, but also looking at the whole profile. Right. So what's the quality of the product and the intended impact, and really having the desired effect. So I think it's a great discussion that we're going to continue to have as more products come through the pipeline, and we see what their impact are.
Absolutely. Miles, can you just show us the results of this latest poll question here? All right. So it looks like in terms of where we want to see more guidance, it's actually a pretty close play here across all of it. So we've got the winner here is analytical characterization at 52%, followed by, it looks like, potency testing or assurance, right, for mRNA products, followed then by raw material quality. and the non-clinical requirements and prior knowledge platform application are kind of rounding out the top. So I'm curious, do you guys have any reactions to that before we jump into some of the more operational considerations we have for working with the regulatory agencies? I don't know, Oriana, if there's any knee-jerk reactions you have. You're the biggest face on my screen right now. So we'll put you in the hot seat.
Yes. Looking at that, I think, again, it speaks to the need for guidance across all aspects of the mRNA technology, right? So there's a lot of different areas of focus and interest. And right now, everyone's really digging in. So I think that's the kind of the results that we're getting across the board. Wanting to have more specification or guidance in that space. So that was very cool.
We have many concerns, many questions, many focus areas. Melanie, any final comments on the poll before we jump in?
Yeah, well, I think I mean the raw material quality piece as we jump into the next one is of interest to me. That's kind of the hot button topic that I've been spending my time on, along with the analytical stuff as well. And we for those on the webinar, the reason that platform, Anna Rose put platform in parentheses is
it's an emerging debate.
Regulators don't like that word. We found out.
They don't. They actually hate it. So an interesting debate emerging about what we call the platform.